now like to gladly move on to the next session, which is meant to start 10 a.m. as of now in a minute, and that would go with the presentation from our Kenyan speakers, Dr. Jilani Ngala and Ivy Mutai. So I would be moderating the session myself and another team member here, Ojedinro Messi. And first of all, I would like to acknowledge the presence of our speakers and also read their research, their summary, their citation. So I would come up to read Dr. Jilani Ngala's citation, and Messi will take up for Miss Ivy Mutai. So the citation for Dr. Jilani Ngala. Dr. Jilani Ngala is a senior research scientist at the Institute of Primate Research, IPR, Nairobi, Kenya, and he holds a BSc in biology across botany and zoology from the University of Nairobi, Kenya, a master's degree in anthropology from the University of Dar es Salaam, Tanzania, and a PhD in neurosciences from the University of Witwatersrand, Johannesburg, South Africa. He also has a postdoc from Newcastle University's Institute of Neuroscience, United Kingdom. His research areas are in non-communicable diseases and virology, specifically bacteriophages and neurovirology. Since 2015, Dr. Jilani started to focus on bacteriophages as early as 2014 and got training on phage isolation and characterization from a workshop organized by Phages for Global Health in 2017 at Makerere University, Uganda. He has ever since focused in isolation of phages for therapeutic purposes, mainly against drug-resistant bacteria of public health interest, affecting both animals and plants. Since then, he has been leading a phage team, the IPR Phage Hunters. And so far, this phage team has published about 15 papers and have isolated phages against E. coli, Staph aureus, Salmonella typhi, Enterobacter species, Enterococcus species, Pseudomonas aeruginosa, and Klebsiella. Dr. Jilani has participated in the two East African phage training workshops, one in Makerere University in 2017 and the second in Kenya 2018, where he was the host. He has also participated and presented in phage research work in several conferences, including the Evergreen State Conference, the Biennial Conference at Evergreen State, the Cayenne Conference in Brazil and Italy, and the SAKIDS Conference in Tanzania 2018. He has received several grants both for his research work and for short-term research visitation or specialized training. His lab undertakes research work as well as training undergraduates and graduate students. And up to now, in phage research, he has successfully trained four MSc students, two PhD, and a couple of about 100 undergraduate students. He works in collaboration with Dr. Toby Nigel of the Phages for Global Health United States, Dr. Benjamin Chan from the Yale University, Professor Matakloki from the United Kingdom, Leicester University, which we had as our main speaker yesterday, one of our speakers yesterday, Dr. Jeanette Neil from Leicester University, also United Kingdom, Professor Elizabeth Kuta from Evergreen State University, and Dr. Kinses from Hungary. In addition, he has also won several professional awards and honors based on his career. So that goes the citation of Dr. Jilani and Messi. If you can unmute now, you can read the citation for Miss Ivy Mutai. I think there is an issue with our network, so I would perhaps just read the citation for Miss Ivy Mutai. So the citation for Miss Ivy Mutai reads that she is a research fellow and bacteriophage specialist at the Institute of
I'm here.
Primary. Okay, you can pick
I'm
it
here.
up then.
Hello.
Yeah, pick it up. Continue.
All right. Miss Ivy Mutai. Okay. Miss Ivy Mutai is a research fellow and bacteriophage specialist at the Institute of Primate Research, IPRT, Nairobi, Kenya. She is a phage expert and consultant with the U.S. Army Medical Research Directorate, Africa. Ivy holds a master's degree in biochemistry from Kenyatta University. She's to graduate in December 2022 and a bachelor's degree in the same field from Karatina University, Kenya. Ivy has been working under the support and supervision of the Director of Research at the Institute of Primate Research, Dr. Atunga. She is the recipient of the Phage Recognition and Achievement Award through the Evergreen State College, United States, and the Institute of Primate Research. This was based on her excellent work for successfully and continuously training and mentoring Phage participants in the Phage Lab Training Program at the Institute of Primate Research and other biomedical research institutions across sub-Saharan Africa.
So thank you. For our speakers, you now have the floor. You can continue your presentation. So we will share the presentation from our hand so you can follow with ease and present.
Thank you so much, the organizers, for giving us this opportunity to share with you what we're doing right here in Kenya, and perhaps have a view of the African continent in terms of what we are doing. It is our pleasure to zero in on what we do at the Institute of Primate Research. Thank you for that very elaborate bios. I appreciate it. So, as we celebrate the World's Phage Day with everybody else, we would start by, in the next slide, looking at our contribution to the AMR as a burden that even the UN has looked at it as a threat to humanity. We are looking at this as a condition that is threatening to wind down the good things that have happened in medicine. And for a start, the Phages for Global Health just came in handy. And we were amongst the first of the cohort to be trained in Africa, so that Africa then joins the rest of the world in getting an alternative to the chemicals or the molecules that have been used as antimicrobials. And we are looking at a longer duration of illnesses if and when we realize that the current regime or current regimen of antibacterial treatment is actually waning. And this is not news, it is what is truth. And in the next slide, next slide, please. We are looking at the burden of the disease and the possible outcomes and fatalities if an alternative is not found. And the statistics would show this perhaps has been displayed to you so many times that it's now becoming so familiar. But realizing that Africa and Asia would be the hard hit of the continents in the world. And so where are we headed to in Kenya? And looking at the African continent in the next slide. We are looking at 2050. It appears like it's so far off, but I mean, this is the situation as it is now. Now, a comparison of one of the conditions that has been with the world, killing as many, causing fatalities and incapacitations, HIV/AIDS. If you look at HIV/AIDS through time, from the time of its emergence on the globe, several mitigations have been done. It was in the rise, and of course, at a time like now, this is kind of like slowing down, despite a few peaks here and there at times. But if you look at AMR, by the time we're discovering it, it has been just a steady rise, meaning in as much as we want, this is what we need to do. Get a cheap, viable, proven alternative to combat AMR and definitely slow down the death. In the next slide, and so looking at the genesis, just going through the genesis of the Phage research in Kenya and at the IPR, the Institute of Primate Research, begins with the Phages for Global Health. It was such a strategy to teach African scientists, which we have benefited. The first was in East Africa, and definitely the next one happened again in East Africa, in Pwani University, and we in IPR were the hosts. Me and Dr. Atunga, having trained in the first, we became hosts of the second in the Pwani University. And throughout this, we have been thinking of the best approaches to increase the number so that we have a good number of Phage researchers. And the first thing that we were looking at is, besides the isolation and characterization and possible use, where were these uses going to be? Perhaps into human and animal, including plants. We realized that being new, this was not going to be an easy entry as an alternative into the human quickly, but the plants and animals that would be. But today, all we know is that health is all-rounder, the One Health approach to things. And so when the beast or the animal is healthy, the human is also healthy, and so is the plant. And so we quickly went into looking at an East African collaboration with Tanzania, Uganda, Ethiopia, and we had a consortium, which then we were looking at the next thing that we need to do is, besides the increasing numbers, we need to look for money to do the research. And as we retreated individually into the various countries, we then were encouraged to do much of the research. And this is going to be illustrated to you later. So at some time, we had an opportunity to be part of the Evergreen, the annual or the biannual meeting. And so Africa was for the first time making its presence in this forum of phage hunters, and of course, aiming towards using phages as cheap, viable alternatives to combat AMR. Now, the schools have increased in numbers until phage training got a hitch from the appearance of this virus called coronavirus. But before the beginnings of coronavirus, we had had a third East African training in Tanzania in 2020. And so what are the outcomes? Networking was one, and we've managed to do that. We were imagining of startups, but that would be a long shot. In the meantime, what we wanted to gain was if we could have webinars, establish hands-on, increase the numbers, get ourselves to isolate as many phages as is possible, and put them in a phage bank with a view to then take that and form libraries, form concoctions, and of course, then going to helping ourselves when it comes to that. And of course, COVID taught us that if Africa was left out in some of these things, when an epidemic or a pandemic hits, Africa is left alone. So in the next slide, along the way came the idea after the East African and of course expanding into West Africa. And the idea was also to go to North Africa as well as South Africa. The AMR group, or the Phage training that was more or less like an AMR group that was an Africa-wide, decided let's have an African forum, an African Phage forum. And within the African Phage forum, in the course of time, we then gave rise to, or we gave birth to the Kenya Phage, which is now at infancy. But eventually what we needed to know from that is within the schools that we had, what was the impact this far? Taking stock of the people that were involved in the Phage, for the Phage research, at what points, universities, research institutions, individuals maybe. And in the next slide, we are looking at the kind of things that the Phage Forum for Africa, the African Phage Forum, was aiming at. And those are some of the things that we were looking at, the various institutions, and we realize a very small number of researchers or researches are ongoing. You also note that there are places that, or there are regions that Phage research has taken off and is flourishing, while others are still picking up. But eventually what we need to do, or what we are aiming to have in this platform, is have an Africa that is ready for the AMR in terms of capacity to isolate, capacity to characterize, and capacity to formulate and then test concoctions or cocktails against any particular disease or condition that we'll be looking at. More so, the entry point and the easy entry point was in animals and plants, but eventually we would go into the animals. But as it were, with the phages isolated this far in the various places, we can be sure with the exchange of phages, the formation of phage banks, regional or country or institutional phage banks, we'll be able, almost ready to combat AMR with more training. In the next slide. So we had various stages of the research in Africa, as is seen there. But so where are we? What do we want to do? What's our future? As I said, we want to get into agriculture, skin products, sewage decontamination, and that basically looking at protection of the environment. We perhaps would want to do that into pests. And the last time us here in IPR were looking at what if we formulated phages that would go into control of locusts that had invaded us some 1 year or so behind or back. And it is basic that the water sources in Africa are highly contaminated. And with phages, that's one thing that we can actually gain with the capacity and where we are this far. In the next slide, it has not been a rosy path for us in Kenya. For us in IPR, we are looking at what we have really gone through. Wading through the donor community for funding in an area that is kind of like emerging in Africa, I would say. This has actually been a challenge. Most donors or donor community or the donor community has funding for particular disease conditions, and that makes entry into formulation of phage cocktails as alternatives really hard, and a consideration for an award next to impossible. The national resources in developing countries, our priorities are much more broader than our resources can match. And more so when we have believed in the one molecule, one target way of doing things, convincing policymakers that this is what we need to do. At times, even sharing the little resources that we have becomes also a daunting task, but we're almost getting there, engaging the policymakers and the internal sources of funding, and hopefully we will be getting there. The lack of knowledge about phages, and when you have something like COVID striking, and you're talking about bacteria and phages and viruses, then somebody then gets to lock in the sense that, isn't COVID a viral infection? So where do you, so we have a lot of explaining to do and engagement to do. So the acceptance, the lab infrastructure, the phage skill sets, which is still building, and of course, COVID also taught us that Africa, without its consumable base, the ability to manufacture consumables for our laboratory settings, led African research and science to be at a standstill for as long as COVID was there. If we were to characterize using TEM, then there are very few spaces that would have TEM, for example, such very expensive machines that a majority of us can't afford, unless the science in these areas or these spaces were organized in national laboratories. That's perhaps my view, that Africa would be able to do most of the research that it endeavors to do. The sequencing and the sequencing platforms. This is a dream to most research institutions, universities, and of course you'd have one or two platforms present in far-flung areas. But with these, we have managed to do something. In the next slide, we were looking at collaboration as part of what we could actually do. But at the end of the day, what is taking much of these collaborations is finding partners, and we want to find partners from within, then partners from within the institution, partners from within Kenya, partners from within Africa. And the birth of Phage Kenya gives us that opportunity. And the call areas for collaborations, again, are skewed to what is it that people want to do. And we say whoever pays the piper calls the tune. And Phage research basically would have the list of a consideration for funding. Next. Now, how far are we? Have we done enough? We are still doing more. My colleague Ivy will take us through the next phase of this presentation, and I thank you.
Good day and good morning, everyone. I hope I'm audible enough.
Yes, you
Yes,
are.
you
You
are.
can go ahead.
Hello.
You are audible.
Okay. So I'm going to present on the status of phage research. Okay. Status of phage research in Kenya, but I'm focusing on IPR, only a few studies that have been done in IPR on phages. Next slide. So one of our studies that was recently done was by Dr. Nutin Fernand, who happened to be one of our PhD students, who was working on a genomic evaluation of novel Kenyan virulent phages isolates. And the host bacterium that was used in phage isolation was carbapenemase resistant or producing. And later on, he did a safety determination of the phage lysates in bulb C mice. So this is because of the occurrence of MDR clade pneumo strains, especially the carbapenemase producing strains, which has become a global healthcare concern, which calls for the need for alternative treatment due to the broad spectrum of antibiotic resistance. So one of his objectives was to evaluate the genotypic features of novel Kenyan virulent phages against carbapenemase producing Klebsiella pneumoniae, and also to determine the safety of their phage lysates using mice model in a preclinical study. So the results of this study, it revealed that when he did a genomic characterization of this phage, it belonged to the genus Slope K virus. And when he did the endotoxin levels of Klebsiella phage lysates in vivo studies or in the mice, he realized or he noticed that the phage lysates still contain endotoxins, which calls for a way of removal or eliminating these endotoxins prior to in vivo studies or phage therapy. Next slide. Next slide, please. Next slide. Also, the previous slide. He also noted that the coding sequences were 85.55. He actually had two novel phages against Klebsiella pneumo, and that one he named them carbapenemase producing resistant SA and SB respectively. The coding sequences were 383 over 483, which indicates it's 84.55% coding sequence as compared to the other one, which is 85.37. So those were the comparative analysis between the two phages. And then another difference between the two phages is that one of the phages contained an autonomous glycyl radial cofactor, which functions to catalyze the metabolic reactions in a low oxygen environment. So which is a plus to this phage such that it can survive in anaerobic conditions. And also after infection of the bacteria and introducing the phages into the mice in the lung, liver, and kidney, after 72 hours post infections in the groups of mice, injected with the phage lysate via intranasal route, unlike those that were lysates that were introduced through intravenous. Next slide. So the annotation features of the Klebsiella phages, the two phages. Another study that we have done at IPR is done by Dr. Joseph Michaels Ochieng, who was doing it for his master's degree. He was working on isolating of lytic phages against methicillin-resistant Staphylococcus aureus in mice.
And these phages were isolated from environmental wastewater. And he was also looking into a solution towards the rise in antimicrobial resistance, especially in the methicillin-resistant Staphylococcus aureus. So this was one of his objectives, to determine how efficient are these phages in mice models. Next slide. So when he did in vivo studies, he realized that the use of
Hello, Ivy. Are you still speaking?
somehow I lost my network on my side, apologies. But he realized, yeah, the graph on the right indicates the treatments of the use of phages. That is use of a control phage. As we can see, the purple line, line graph indicates control phage. That is a phage with no bacteria, and we can just focus on the green the green curve, which indicates the presence infection with the multi-drug resistant Staphylococcus aureus and the phage, which is more effective as compared to multi-drug resistant, the presence of the bacteria, the presence of the antibiotic plus the phage. End of it all, we can conclude that the use of phages alone are more effective in this study as compared to use of a phage and an antibiotic as well. Next slide. So other related studies on methicillin-resistant Staphylococcus aureus were also still done by Dr. Joseph Michael Ochin, which were experimental phage therapy against hematogenous multi-drug resistant Staphylococcus aureus in mice. So with these two studies, he was trying to compare the effectiveness of these phages in different conditions. And they are still associated with the phages isolated in the first study as well. Next slide. On this slide, we are focusing on phage training reaching East Africa. This was after Dr. Betty Carter visited IPR in 2015. And that is when Dr. Joseph was doing his master's project on phage. And that is when the phage training in IPR started. So, but for the training to go on, the donations were made by Dr. Carter Elizabeth, which we are highly appreciating her support. And following the phage training, there was also organization of a workshop by the Phages for Global Health. That was the second East Africa that happened at Kilifi. That is Pwani University in Kenya. And after that, the phage group at IPR became so vibrant until we established our own phage club and continuous phage hands-on training on phage training and creating awareness on antimicrobial resistance. So far, as Dr. Gilani had indicated, that we have trained 4 MSc students, 2 PhD students, and over 150 undergraduate students from different universities and research institutions. So, and this training has been impactful by the trainings that have been led by Dr. Toby Nagel of Phages for Global Health and her team. Next slide. So another study that was done or was led by Dr. Atunga Nyachio, this is when he had gone to Uganda, Makerere University, Professor Jessica Nakavoma's lab. That is where they isolated and characterized novel lytic phages against multi-drug resistant E. coli and Salmonella typhi. This is because these pathogens are zoonotic diseases. That it means they could infect humans, livestock, and wildlife. And the cause of these Salmonella typhi causes over 21.7 million illnesses, and the death rates that's high, which are 217,000 deaths annually. So the objectives of this study were to isolate lytic phages against Salmonella typhi and E. coli, and to evaluate the host range of the isolated lytic phages, and also to establish the stability of these phages in different pH and temperature of the next slide. So on the results section is that they isolated phages against 34 bacterial isolates, but they had to do criteria of which isolates are they going to use to isolate the phages. So there were 23 E. coli and 11 Salmonella. So 2 were enterohemorrhagic E. coli, and 5 from environmental sewage chicken, and the Salmonella were from chicken, Marabou stock, tatters, snake, and gorilla. But now, upon subjecting them or testing them for antimicrobial susceptibility, they were looking for the resistant strains, which is indicated below, that is as S and R. And they later found out that E. coli had the highest prevalence of resistance of 52%, followed by Salmonella, which had a resistance rate of 18%. But most of the strains were resistant to sulfamethoxazole, and this was from actually high resistance was observed in chicken. So since most bacteria were resistant to one or more drugs, it indicates that these strains were resistant. They were multi-drug resistant strains. Next slide. So this is still an explanation of the previous slide of antimicrobial susceptibility test. E. coli still had high prevalence, followed by Salmonella, and high resistance was observed in sulfamethoxazole. So this is still an explanation of what I did in the other slide. So next slide. So when they did host range, so amazingly, they got the most potent phage to be from chicken. And that means from this study, they found out that samples from the chicken had the highest resistant, but also gave the most potent phages. So chicken had the highest host range. That means they lysed the different strains of bacteria that were tested. That is for the E. coli phages. So it lysed all the strains that were tested, unlike
Apologies for that. I think it's the speakers. It's a flaw in the speakers network, so she should be back on anytime soon. Hello, Ivy.
Hello, this is still Ivy. I have technical problems on my side.
Okay, is that still Ivy?
Yes, this is still.
All right, you can go ahead then, no problem.
Me. Apologies on my side. Potential sources of phages for therapeutic purposes. And Salmonella phage only killed the host, so that means it did not have a wide spectrum of activity. Next slide, please. Next slide. So on temperature variation, the phage from the chicken was subjected to different temperature variations, and
they found out that 37 degrees is where the phage gave the optimal efficiency, And apart from that, there was still efficiency between 4 and 59, 59 degrees centigrade. So that means this phage is potent and can be used across different temperature variations. Next slide. So in terms of pH variation, the same phage was subjected to different pH variation. And from in the graph on the right, it is visible that it is evident that the phage had optimal activity at pH 7.4, which is basically neutral. And this indicates that this phage can actually, if you want to use this phage for therapeutic purpose or biocontrol, that means the pH 7.4 is the optimal that could be used, but still it's still efficient across the from 4 to 9.4. Next slide. So another study that was done by one of our MSc students he was trying to isolate phages against multi-drug resistant Pseudomonas aeruginosa. And this is because Pseudomonas aeruginosa actually has caused a lot of morbidity and mortality rates currently. And the emergence of these bacteria, the emergence of resistance against these bacteria calls for an alternative treatment such as phage therapy. So in this study, he isolated these phages and tested them in mice. And then he characterized them morphologically and also tried to assess the effect of in vitro growth kinetics, host ranges, and the stability also of the phages in different physical chemical properties. Next, please. So on term analysis, the Pseudomonas phage showed that this belongs to Myoviridae family. So it has an icosahedral health and approximately 50 nanometers. So this indicates that basically this phage belongs to Myoviridae family. Now, next slide. So he also did the stability studies on different temperature and pH. And as we can see on the right, we see that the phages on temperature were efficient from 4 to 50 degrees, but they were inactivated beyond 51, that is 60 and 80 and degrees centigrade. But in terms of pH, we can also capture that at pH 7, this is where the optimal activity of the phage was also observed, which is in line with the other study that was done by Dr. Atunga in Makerere University. Next slide, please. So in terms of one-step growth curve, he did this one-step growth curve trying to optimize method of use of a phage against in vivo studies prior to that study. So these indicators that the latent period of the phage was 15 minutes. That is the period of infection and replication was 15 minutes, and burst size of the 316 viral particles per infected cells. So that could mean that this phages was producing a very good burst size for replication cycle. And also, next slide, please. Next slide. So he also did the bacteremia and viremia levels to compare the effectiveness of this phage as compared to antibiotic plus the phage to see how effective when you compare the two of them. But you can see that the one that has a phage, it has a better activity as compared to the one that has better activity as compared to the one that has combined treatment. Next slide, please. So the other study, which was done by Ivy, who is me, was efficacy of diversely isolated lytic phages against MDR and Enterobacter cloacae isolates in Kenya. This is because Enterobacter cloacae has been associated with hospital-associated infection, and it's the rise in MDR, and it affects the patients who are either immunocompromised and later on leads to sepsis. So the studies, the objectives of this study were to isolate and evaluate the antimicrobial susceptibility pattern of the MDR E. cloacae from the environment, and then to evaluate if the phages against these MDR Enterobacter cloacae is present in our environment, which is environmental wastewater, but focusing on the informal settlements in Kenya, and also to evaluate the efficacy and stability of lytic phages against MDR, against the different physical chemical properties, and also to do a host range study against a panel of MDR isolates, and to carry out transmission electron microscopy of the most potent phages from host range. Next slide, please. So in host range analysis, it was found out that the E. cloci phages that were isolated showed cross-reactivity against all Enterobacter cloci isolates, which were 30. So, and the most potent phage that is highlighted in red was the most potent. It lysed 67% of the bacterial isolates that were tested, and the least potent isolate, the least potent phage lysed 27% of the strains. It's the least in this study, but maybe in another study, this could be the highest. So these indicates that the phages that were isolated against Enterobacter cloci had high or wide host range. Next slide, please. So in terms of, this was a comparative study to compare the susceptibility, how susceptible were the bacterial strains that were isolated, that were subjected to host range to the phages. So the ones that were highlighted in red, this indicates that these isolates were 100% susceptible to all the phages that were tested. So yes, so the 23 bacterial isolates were susceptible out of the 30, while 7 isolates were not susceptible to any phage. So this calls for looking for phages against the 7 isolates that were not susceptible to any phages. Next slide, please. So on the effect of storage or phage activity on the phages, we found out that the most, the optimal phage activity or the lytic activity was observed at 37 degrees, followed by 4 and minus 20. This was after storage after 2 weeks. So you can see that if you're using a phage for therapeutic studies, it means human beings are sorted because of the human body temperature. But at 4, there is a bit of development of resistance. So that means you could not store your phages for long at minus 4. So you proceed to storing them for long-term storage at minus 80. Minus 20, the phage activity, the phage titer or activity decreased, and this is because of the crystal structure of the ice actually degrades the phages. Next slide, please. So time analysis of the most potent phage was isolated in one of our informal settlements, and it has been captured there. It has an icosahedral head and a contractile tail. And this shows that this phage belongs to Myoviridae family and the Codovirales order. So apart from doing this stem analysis, this phage has also been, genome sequencing has been done on this phage to also support the taxonomic classification, but the phage sequences are still in press. They have not yet been released. Next slide, please. So from all these studies that a few studies that have been done in IPR, it shows that our environment are rich sources of novel lytic phages and can be used in treatment of these superbugs or the MDR, such as or pan-drug resistant Staphylococcus aureus, Pseudomonas aureus, E. cloci, Salmonella typhi, and E. coli. And we have also seen that the phages are stable and efficacious in broad pH, that is pH 5 to 13. And the thermal stability range is 4 to 60 degrees, and a short latent period and a wide host range. So this means if we are proceeding to product development, these are the factors to consider before development or developing a phage product. So, and also this supports the fact that phages could be used as alternative to the current antibiotics failure or the inability of bacteria to respond to an antibiotic. So, and again, further research is needed to ensure that more phages are isolated, genetically characterized, and commercialized for therapeutic use. And again, it is brighter. We are in it despite all odds. It's always darkest before dawn. So we are somewhere as Africa, and the sun will come out. Next slide, please. So acknowledgements, I would like to thank all authors
of the outlined phage research for taking their time to do this research. And so Phages for Global Health, Ibadan team, the African Phage Forum, and IPR, and also the participants who have managed to attend the World Phage Week. Next slide, please. Next slide. So for further inquiries and call or collaborations, these are our contact details as our team in IPR, but others have already left, like Mutai is in Benin already, but feel free to contact. These are the contact details. Thank you very much.
Thank you very much, Ivy. Thank you, Dr. Ngala. That was a very nice presentation. At least I must say that it portrays phage research in Kenya. It gives a very elaborate analysis of what you've been doing so far in IPR, and that's a very fantastic job. So before I go ahead, if you have any questions concerning the presentation by Ivy and or by Dr. Ngala, you can just drop the question in the comment box, or you can signify by raising your hand. And while I wait for that, I would like to acknowledge the presence of another faculty member here who also presented yesterday, the person of Dr. Temitope Faleye. It's nice that we have you staying around with us despite the time zone difference that's and your busy schedule, which I know is not very friendly. That will be around like 2 a.m. over there. Thank you very much. So if you have any question, you can drop it in the comment box. But I would proceed with my own question, Ivy or Dr. Ngala. So I have this question to ask, which is that in IPR, perhaps you might have hinted it back in the presentation, and I just didn't quite follow. Have you done any clinical tests or clinical experiments to look into phage research capacity in IPR? Have you done anything about clinical therapy? It could be in animals or sorts, but has there been any research carried out concerning clinical therapy of phage use?
Thank you so much, Daramola, for your question. For clinical trials, we have done only in the lower animals, that is mice, and we are looking forward to trying or doing studies in higher animals such as baboons, which are closely related to human beings. On the part of human clinical trials, there is a challenge because the use of phages in Kenya has not been implemented in the use of in human health. So that is what we are trying to channel currently. At least we see a way forward after which we can try to venture into human clinical trials. Thank you.
Thank you very much. Okay.
If I was to add on to that.
All right. Yeah. Go ahead, sir.
We are a preclinical research institute here, so we normally do preclinical research, the use of animals. We realize phage research and then the resultant cocktails that were going to be therapeutics would need to be put into the mainstream regulations. So from the word go, as we were training in our training workshops, we are trying to engage policymakers so that now we entice them to understand what phages are and what their capacity and potential in terms of the emerging AMR issues are. And so at some point, part of what we needed to do was then engage the regulator, which is the Pharmacy and Poisons Board, to come on board as we are doing capacity building, as we're doing most of the experimental work that you've seen, as we also try out those ones in animal models so that then it becomes like this really works. But going into the clinical trials would mean such cocktails are formulated, are to an extent okayed by the Pharmacy and Poisons Board. And then those can now go into the clinical trials. But that's a long shot. So in essence, what we wanted to do was part of what anybody can get convinced quickly is when you start with, say, plants, start with fruits, like phages that elongate shelf life of various fruits, say tomatoes, and then get to the animals. If you can restore animal health from the preclinical experiments, then that gives confidence in these people to believe the cocktails really work and the phages are actually a good shot, cheap relatively, and of course, the best alternative that there can be apart from the molecules. So that's our direction. We want to do the research as we take along the policymakers, as we do the advocacy. So at the end of the day, we have good acceptance when we finally get to now formulation of cocktails after all that. So you've seen that we have done the isolation characterization. We've gone even to genomics. And at one point, we had preclinical mice studies of the Staph aureus that Joseph Ochieng did. So we're headed there, but we don't want just to go there as scientists. We also want to get the policymakers that eventually when we are out there saying this is the cocktail that works, we are with them.
Right, right. I have to agree with you, Dr. Ngala. That's a very good point, and it really addresses that we still have a long way to go in the scope of Africa, as Ivy also reiterated in the end of our presentation. So we have another question, which I believe most likely has been answered already, but I would go ahead to just read again. So from we have a question here. It says, "Nice presentation. Do you guys engage in research as it relates to phages in the development of viral vaccines?" So just in about a minute, if you still have anything to say regarding that, you can go ahead.
Yes. Other viruses other than the phages that we're using, or that generally all viruses, as it were?
Hello.
Hello?
Yeah, okay. I mean all viruses actually. Yeah, all viral vaccines, because I know that phages they are much more safer because they can't infect human cells, and then they can be used as vectors to actually carry the antigen of some other viruses for vaccine development. So I'm just trying to ask if you guys engage in those research.
Yes, thank you. We have started moving in that direction, but of course, we have gone into viral researches such as HIV, of course, with the use of SIV in animal models, and we have an SIV model of HIV. When Corona struck, we entered into helping governments do the testing, the mass testing. And then when it went off, we were looking at possibilities of doing the routine, normal, typical viral vaccine development, but we have not done that yet. We are still in the planning phase. We have turned, ever since that COVID-19 testing and research center, into kind of like a One Health Center, where we want to look at the approach that is the global trend now. Both animals and humans, including the plants, must be healthy for a healthy world. And so we are looking at emerging conditions, mostly targeting the viruses, because we've seen how hard we can be hit with the viruses. So yes, we are more into viral research, but we haven't done much towards development of vaccines, or a vaccine that we can say this we developed from the viral research that we have been doing. Thank you.
Thank you.
Thank you very much, Dr. Ngala, for addressing the questions we've posed so far. So we just have one more slot if you have any question, and if not, we'd go ahead to wrap up the session. But to mention in particular, I would like to appreciate that I would like to appreciate Ivy Mutai, and I would like to appreciate Dr. Atunga and Dr. Ngala for giving us this presentation about their work so far in IPR. That's very welcome, and that's a very informative session. So if we have no question, thank you once again, Dr. Ngala. Thank you once again, Ivy Mutai. And we hope that we would get to catch up later on your work in IPR and in the coming months and in the coming years. And also to all participants, to our faculty members, to the students with us, and to all Phage enthusiasts and Phage researchers that have joined this meeting so far from the start of it yesterday up until this moment. We say thank you very much for joining. Thank you very much for celebrating the annual World Phage Week with us. Thank you for tuning in to join us with this conversation and this webinar series. We so much appreciate that, and we are glad to have your support anytime and every time. So with this, I think we will be wrapping up. Okay, Kibet is nice presentation. What can you say with regards to phage and microbial evolution? I think this question might be much more elaborate than we can accommodate for the moment. But Ivy, can you like succinctly approach this question?
Okay, thank you, Kibet, for your question. In regards to microbial evolution, so as bacteria are also evolving, so phages have also developed a mechanism to be at par with evolving with the bacteria. So if you don't get a phage against a specific bacteria, you go out back to the environment, you'll get a phage that has actually evolved to catch up with that bacteria that has evolved.
Very good. I think that answers the question, Kibet. All right. So yes, that's very good. So thank you very much once again. Thank you all. So this would be a signing off from our end in celebration of the International World Phage Week by our webinar, our annual webinar series. So thank you all for tuning in. Thank you all for joining the webinar series, and we do appreciate all of you present. As we've mentioned earlier, we have our second issue coming up soon, and we will communicate that to you as soon as possible. Williams.
Yeah, yes, thank you very much for this opportunity. I just want to chip in something that is also important. In the session yesterday, Mata Kloki emphasized the importance of putting our voice out there and ensure we get mentors and people that are actually interested in what we do in our circle. So on that note, I would like to enjoin everyone to actually put whatever they've learned in this particular webinar series to share them on social media. And you can definitely tweet at us on Twitter. We're also on LinkedIn, though not as active, but we'll try as much as possible to react and engage with every tweet or every post that you're making. So it will also go a long way in helping us with our brand and letting people know that this is what we do. So that's just what I want to add. Thank you very much.
Right, thank you, Williams. And that's a big chunk addressing science communication, and would very much appreciate that we publicize as much as possible what we've learned about as regards this webinar series. So that's just what Williams was trying to reiterate. So this would come to a conclusion, and we'll be ending the session. The recording will be made available as soon as possible for public access, and we would email that